Good afternoon. Welcome back to Virology. Okay, today we're going to talk about reverse transcription and integration. By the way, we have a guest today from the University of Puget Sound in Tacoma, Washington, Mark Martin. Anybody from Tacoma here? He's a biology professor, and he does one of our podcasts called Matters Microbial. On every slide, the first slide, I have quotes, and I know that you don't read them at all because you don't need to read them to pass or get a good grade. But this one is pretty cool. The quote from Alice in Wonderland. "One can't believe impossible things," said Alice. "I dare say you haven't had much practice," said the Queen. "Why, sometimes I've believed as many as 6 impossible things before breakfast." So that's totally suitable for this lecture. A lot of impossible things are going to happen today. So this is a little introduction to tumor viruses, which we'll talk about in a separate lecture about how viruses cause disease. This is a discovery of tumor viruses and how they work. So this starts in 1908. Chicken leukemia virus is discovered. So a virus that causes leukemia of chickens. 1911, Rouse sarcoma virus by Peyton Rouse here at the Rockefeller, at the time, Medical Institute. It took 55 years for him to get his Nobel Prize, the longest incubation period ever. And they were called tumor viruses because they called tumors, although the leukemia was not considered to be a cancer because it wasn't solid. And years later, people realized not all tumors need to be solid. They can be cells that are growing improperly. And these are the people that discovered these viruses, tumor viruses. Then they were found to have RNA genomes. Really interesting. In 1911, actually it was the 30s, they're found to have RNA tumor viruses. Then along comes Howard Temin many years later, and he says, okay, these RNA tumor viruses cause permanent changes in cells. That's what causes a tumor, as we'll see when we talk about transformation and oncogenesis. So there's no way that RNA can do that. So it must be that the viral RNA is converted to DNA, which then integrates into the host and becomes a permanent part. And this was his provirus hypothesis, the idea that these RNA tumor viruses had some kind of enzyme that made a DNA copy of the RNA genome and integrated into the cell. Really, really brilliant insight, actually. At the same time, David Baltimore, who you remember from the Baltimore scheme, you remember how many different viruses are in the Baltimore scheme, Mark?
That would be 7, Vincent.
That's right, because of the subway sign, right? My podcasting colleague, who unfortunately passed away 2 weeks ago, I always used to ask Dixon, how many different genome types are there? He would say 7, Vincent. Baltimore found that plus-strand RNA viruses don't have the polymerase in the particle, as I've taught you. Minus-strand RNA viruses do. So it took Temin's thinking and said, these viruses must have an enzyme in the particle to copy RNA to DNA, because we don't know of any enzyme that does that. So he didn't come up with the provirus hypothesis, but he said, this is biochemically what has to happen. Okay, so they both published their findings in the same journal at the same time. Discovery of this enzyme called reverse transcriptase. So there's Temin's paper, RNA-dependent DNA polymerase in Rouse sarcoma virus. So he took the virus that Rouse had discovered, and then on the right, reverse transcriptase. In tumor viruses, he used the mouse tumor virus. So the story here is interesting. I spoke many times with David Baltimore about it. He said, once Temin had put out this provirus hypothesis, Baltimore said it took us about 2 weeks to do the right experiments, because he was set up to do polymerase assays, right? He'd been doing all these RNA virus polymerase assays. And he said, Temin had been slaving for years. So he called him up and he said, Howard, we're going to publish this paper soon. And Temin got his act together and did the experiments in his lab, and they both co-published. This would never happen today because you want to publish first. And it's too bad because they both did it, and they both deserve credit. Anyway, they both got the Nobel Prize in 1975 for this discovery. Really important. Reverse transcriptase is a huge enzyme. Huge. You can't imagine how huge it is. So the name comes from the fact that they apparently reverse the flow of genetic information. It's normally DNA, RNA, protein, right? So they take RNA and make it into DNA. And you know, when Francis Crick, who supposedly came up with this dogma, DNA, RNA, protein, they said, what do you think about reverse transcriptase? He said, I never said it was a thing. I never said it was a dogma. So that's what people do. But this enzyme has revolutionized molecular biology. I'm willing to bet that most of you have worked with RT at one point or another in the laboratory. Even Mark Martin probably has worked with it, right?
I certainly have.
You've done some RT-PCRs?
I have indeed.
There you go. So RT-PCR, you take reverse transcriptase, you make a DNA copy, and then you do PCR. In the early days of COVID, every COVID test based on PCR was using RT-PCR. There's some tubes of RT, you can buy them. But of course, you can clone genes, you can make RNA into DNA and clone the genes. You can do all kinds of things. And neither discoverer patented the enzyme because they felt it was their contribution to science, society, in fact. And that would never happen today. Right? Companies would be all over it because you can make tons of money with this patent. So what viruses have reverse transcriptase? The retroviruses, group 6 in the Baltimore scheme. They have RNA genomes, and they convert them to DNA. And we'll talk about this scheme in a moment. But there are also the hepatitis viruses up there, the group 7, which weren't discovered until Baltimore had already put together his scheme of his Baltimore scheme. They have reverse transcriptase in their reproduction cycle as well. And then other viruses related to hepatitis B, as we will see. So back to Rous sarcoma virus, which eventually was called a retrovirus after the discovery of reverse transcriptase. So they were first called tumor viruses, RNA tumor viruses, and then when they found that there's reverse transcriptase in them, their name is changed to RNA tumor viruses. They're enveloped viruses with a spike protein in the envelope so they can attach to cells. And they have within them a nucleocapsid because the RNA genome is encased in an icosahedral capsid. That's shown in red. And the RNA genome, there are 2 copies of the RNA genome. We'll explore why that is in a moment. And then a variety of proteins, including reverse transcriptase, is in the particle, right? Because you need that. The RNA gets in the cell. There's no reverse transcriptase to copy it. Actually, there is, found many years later, but that's another story. We'll talk about that a bit. There's also a protein called integrase, which integrates the double-stranded RNA product into the cell genome. So that's the basic overview of a retrovirus. Here's the genome. I want to point out some features. So the genome in the particle, of course, is RNA. It's a plus-stranded RNA. It's capped at the 5' end. It's polyadenylated at the 3' end. And there are 3 coding regions, GAG, POL, and envelope. The GAG encodes the structural proteins. We'll see more about this later when we talk about assembly. The POL encodes the reverse transcriptase and integrase. And then the envelope encodes the spike. And this is extensively spliced in cells to get you the envelope proteins because they can't be accessed by translation of that mRNA. Now, when the RNA is converted to DNA and it's integrated into the host cell, that's shown up here. That's called proviral DNA. That's a very specific word, which means integrated retroviral DNA. And when it's integrated, it still has GAG, POL, and envelope coding regions, but it also has, on either end, what's called an LTR, a long terminal repeat, which plays a big role in the reproduction cycle, as we're going to see in a few moments. So here's an overview of the reproduction cycle. The viruses bind receptors on the cell. the viruses fuse at the plasma membrane. The nucleocapsid is deposited in the cytoplasm. And then reverse transcription happens in the cytoplasm. So that double-stranded DNA is made in the cytoplasm, and then it passes through the nuclear pore and integrates into cellular DNA. We'll talk about that process in some detail. And then viral mRNAs are made, which are exported and give rise to structural proteins and spike proteins and so forth. And a little bit of unspliced RNA is exported to put in viral capsids. And we talked about the mechanism for getting unspliced messages out of the nucleus. This is one case where you need to have that. And next time, I believe we're talking about assembly, so we will finish the reproduction cycle, talk about how these viruses are assembled. All right, reverse transcriptase. Let's dwell on this a bit. It is a primer-dependent enzyme. The primer can be DNA or RNA. The template can be DNA or RNA. So you don't have to remember anything. It can be either one for this one. And only dNTPs are incorporated. So it only can make DNA. It doesn't make RNA. So even though it can copy RNA, it's not going to make RNA. It's going to make DNA when it does that. And just like all the other polymerases we talked about, the enzyme copies the template in a 3' to 5' direction, and synthesis of new bases are in a 5' to 3' direction added to that primer. We're going to look at this process in great detail because it's fascinating. It's part of what the Queen was talking about in Alice in Wonderland. Now, what about reverse transcriptase? Is it really unique to retroviruses and maybe other viruses? No, it's actually not. After retroviruses were discovered, it was found that bacteria, archaea, and eukaryotes all have reverse transcriptase activity. And so this RT is ancient. It evolved before the split of archaea, bacteria, and eukaryotes. And in case you don't know, this is a phylogenetic tree of life showing the 3 domains: bacteria, archaea, eukaryotes. They had a common ancestor. So that somewhere between the split and the common ancestor arose RT because they all have it in their genomes. In fact, we think that reverse transcriptase might be the bridge between the RNA world and the modern DNA world. So remember, there was a world where just RNA molecules existed. They replicated. They could splice themselves. Maybe they could even make proteins. And then, among the proteins made, arose a reverse transcriptase. We think that RNA polymerases were the earliest enzymes to be made, and perhaps you don't need many amino acid changes to change an RNA polymerase into a reverse transcriptase. So it may have arisen and then copied the RNA into DNA. And that would allow for cellular life to arise because to make a cell, you need a big DNA, and you can't do that with RNA. So this is the idea that RNA is really ancient, and it was the bridge that created the modern DNA world. So here, so the RT is also found in 2 other viruses, hepatitis B virus and Colimoviridae. These are viruses of cauliflowers. And next time you eat a cauliflower, you can think that it has RT in it, but it's not a problem. I mean, you're full of RT. So here on the right is a circular phylogenetic tree of reverse transcriptases. And so here we have viral reverse transcriptases. The cauliflower, the retrovirus, Hepadnaviruses. There's also a reverse transcriptase in an element called gypsy, which is virus-like. And then we have line elements, which are present in eukaryotic cells. We'll talk about what those are. Long interspersed nuclear elements. And then we have telomerase reverse transcriptase. Remember, the ends of our chromosomes have an enzyme, reverse transcriptase, associated with them so they don't degrade and make us age prematurely. And then bacteria and archaea have their own reverse transcriptases. So this is quite interesting. And it's misinterpreted because some scientists a while ago, after the mRNA vaccines for COVID came out, they did an experiment where they put the mRNA vaccine into a cell line that was overexpressing one of these line reverse transcriptases. And it was copied into DNA. Of course, it's going to be copied into DNA. It's a reverse transcriptase. You put RNA, probably every mRNA in the cell was also copied. But they said, aha, these mRNA vaccines can integrate into your genome. Did that experiment show that? No. But, the science breakdown in the press is really a problem. They don't follow their experiments through. There are no controls. Anyway, that's the basis for that. And you hear it till today. It's still being discussed that these vaccines are bad because they can integrate. And there's zero evidence for their integration, okay? I want you to understand, zero. And millions of people, including yours truly, have gotten mRNA vaccines. I'm okay. There's no evidence for integration. And they went through a lot of safety tests to rule that out as well. So RT is one of the 4 nucleic acid polymerases that we've talked about. DNA-dependent DNA polymerase, DNA-dependent RNA polymerase. Here's the reverse transcriptase and poliovirus polymerase. They all look like a right hand with the active site in the palm of the right hand. For HIV-RT, the thumb and fingers do not touch. So it's an open active site, which is down here. And just like all the other polymerases, RT, RNA-dependent DNA polymerase, has sequences in common with the other polymerases, particularly this active site here shown in yellow. And in the active site, remember, there are 2 metals that are coordinated by 2 aspartate residues, and there they are, right there in the yellow sequence. Now, RT has a second enzymatic activity associated with it. It's not just one enzyme. It's called RNase H. RNase H is an enzyme that cleaves RNA when it's in duplex form. That means when it's double-stranded. It could be RNA-RNA, or it can be RNA-DNA. And you will see why this exists in a moment when we go through reverse transcription. It makes endonucleolytic cleavages. It means it cleaves internal. If it cut at the end, if it chewed in the end, it would be an exonuclease, okay? And there are plenty of those, but this is an endonuclease. And it makes short oligonucleotides with a 5' phosphate and a 3' hydroxyl. It doesn't really matter because all it's doing is degrading the RNA template, as you will see. But that's its function, because as you're copying the RNA into DNA, you no longer need the RNA. So RNase H gets rid of it. Here's the structure of HIV-RT. This happens to be a dimer. Not all of them are dimers. 2 subunits called P66 and P51. And at the top, they're shown together. But what I want to show you here is the relationship between RNase H and the active site, which of course is in the palm domain. So here's the palm domain, and then a bit downstream is the RNase H. Here is a RNA that's come in the active site. It's been copied to DNA, which would be in blue here. And then as it's passing RNase H, the RNase H is going to chew up the RNA. It's not really shown here, but in the next slide, I'm going to show that to you. So basically, the polymerase makes the DNA copy, and then the product passes the RNase H domain, and that chews up the RNA. And that's shown here in a cartoon. So here is our polymerase. The active site is here. There are 2 metals that are coordinated by the aspartates that are needed for catalysis. And the RNA comes in here at the left side. It passes through the active site. It is copied into DNA, which is blue. So now you have a duplex flowing across the surface of the enzyme, and then it passes the RNase H site, which also has 2 aspartates coordinating 2 metals. So even though it's degrading the RNA, it's by a reverse mechanism using 2 metals. And then out comes single-stranded DNA. So it is beautifully designed. So you make the DNA in one end, and then at the other you degrade the RNA. And just for your reference, here's the thumb domain, and here are the fingers. You can see it's open. It's not a closed configuration like poliovirus. So How fast does this happen? It takes 4 hours to copy one viral genome. There's no speedster here, right? It takes a long time, and it's very error prone. It makes a mistake in every 10,000 to a million bases. And that's relevant for HIV. There's a huge diversity in HIV. Within a patient, the virus undergoes mutation to change and avoid antibodies. It's because of that error rate. All right, our first question. Reverse transcriptase has revolutionized molecular biology. Which statement about the enzyme is not correct? RT is unique to retroviruses. RT is packaged in the particle. RT has RNase H activity. The name of the enzyme comes from its ability to reverse the flow of genetic information. Might have bridged the ancient RNA world and the DNA world. So which one is wrong? Okay, Mark Martin, which one is wrong? A. Oh, I shouldn't have said that. That's so nobody else would know. Yes, A is wrong. Let's see. Most of you got A. RT is unique to retroviruses. Of course, it's not. It's packaged in the particle. That's correct. So that's the only other one that people picked. But just 2 people, because 22 of you answered. So I'll show you why it's in the particle. I showed you right here. Reverse transcriptase is in the particle, that little blue circle. Why does it have to be in the particle? Because even though cells have reverse transcriptase, it's actually in the nucleus. And RT occurs in the cytosol, so it's not accessible. But we used to think that RT was unique to retroviruses. And so, but in the cytosol, there's none for the virus to utilize for reverse transcription. All right, we're going to talk about reverse transcription in some detail. But first, the RNA. In the virus particle, there are 2 copies of the RNA. So we say it's diploid. And the RNA is coated with nucleocapsid protein. And there are about 50 to 100 molecules of reverse transcriptase for virus particles. So when we talk about the process of a reverse transcription, this is happening on 2 RNA molecules, but there are lots of RTs that are working on those 2 RNA molecules. These 2 RNAs, of course, there they are, capped polyadenylated GAG, Pol, envelope coding regions. They are hybridized at their 5' ends, so they stay together. And in the virus particle, there is also a tRNA bound to each viral RNA. And that red part is the primer binding site. It is not a radio station. It is a primer binding site. And that's where the tRNA is bound. And you'll see why in a moment. Here is on the right how these 2 RNAs stay together. So the 5' end of the genome is highly structured, as you can see. And this S1 loop in particular, as shown here, bases within the S1 loop can base pair with an S1 loop from the other RNA. And so basically, when you start with 2 RNAs, these sequences base pair. Isn't it cute? They call it a kissing loop complex. But they're basically hybridizing to each other. And that keeps them together in the virus particle. And you'll see the implication of that later when we talk about assembly. All right, so that's what's in the particle. Why are there 2 molecules? This is an interesting problem. This is what we think. We think it makes the viruses resistant to mutation, like UV and ionizing radiation, which viruses encounter, right, as they move around the planet. So they have 2 copies of all genes, so that if one is mutated, they have another one that they can use to rebuild a functional genome. So here, for example, we have 2 genomes which are where this gene A is mutated. It's red, so that means it's mutated. And the polymerase, when it copies these 2 genomes, it randomly copies bits of both RNAs. Just randomly goes back and forth. So there's no logic to it. It goes back and forth, and eventually it will make a functional genome when there are mutations in one strand. So that's the idea, that it makes these viruses relatively resistant to UV. To which I say, no other virus does this. So what's the story? Why does this virus have to do it? Or what's the function? So that's the real question. What is it doing in this virus that it can't do in others? I don't know. So this is the primer tRNA binding to the retroviral genome. I just want to show you where it is. There are the two RNAs with the primers bound. The tRNA binds to the primer binding site. It's not very far from the 5' cap. And on the bottom here is showing you in some detail how the binding occurs. You can see the length here. Here we're showing base 56 to 130, so very short sequences. Here's the viral RNA here. That's the primer binding site. And here's the tRNA, and it kind of insinuates itself into the primer binding site. It actually hybridizes with two parts of this, two parts of the stems of this stem loop structure. And for each retrovirus, there's usually a specific tRNA that goes in here. So for ASLV, which is basically a leukemia virus, it's a tRNA tryptophan. So I told you that DNA synthesis occurs in the cytoplasm. So here's what happens. The virus binds its receptor, fuses at the plasma membrane. The capsid becomes permeable so that required cofactors can get inside, and then you make a double-stranded copy of the genome. So I want to show you how this double-stranded copy is made. It's amazing. It is amazing. I'm not showing it to you to confuse you, because for the exam, you can look at these pictures. I want you to understand, though, how it happens. So here's the RNA coming in the cell, and it's positive stranded. Here's the 5' end, and here is the 3' end. So we have here the tRNA bound, and if you look at it, you go, why is it bound so close to the 5' end? Why doesn't it bind to the 3' end? Aha, there's a logic to this madness, as the Queen might say. So the polymerase uses the tRNA as a primer, makes DNA complementary to these sequences. And these are very important sequences. Little U5 are going to be copied to big R, big U5, okay? RNase H degrades the RNA template. So how can you go anywhere else? Well, there's the 3' end of the genome, which has little r, which is complementary to big R. This is not just one base, it's a series of bases. So they can easily base pair, and the enzyme can continue copying from the 3' end. So it's a template jump, right? You'll find out why it does that in a moment. So here's the template exchange, the annealing of the big R and little r, and then the polymerase continues to make DNA till it goes all the way around. And the RNase H is working at the same time, except it leaves a little piece of RNA called the PPT, the polypurine tract, which is going to be the primer for the other DNA strand, because you need primers for both strands. You'll see that in a moment. So we almost have a full first strand here. It's still attached to the tRNA. Here we are, and we finished copying that first RNA. We also copy the primer binding site, right, which would be where the tRNA is going to hybridize. And you see that the synthesis of the second strand has already begun, the light blue color, using the PPT as a primer. All right, the primer is taken off, the tRNA is taken off, and now you have the full first strand, and now you can make a full second strand from that. And again, the ends are juxtaposed by these complementary PBS sequences, which are present on the first strand and the second strand. So that's where we are there. My plus strand DNA continues, and we now have a full-length double-stranded DNA. And look what we have done. We've made two LTRs. The LTR is composed of U3RU5, U3RU5. So this jumping of the template exchange has basically put two LTRs on the DNA because it wasn't on the RNA. as you see here. The RNA doesn't have 2 LTRs. It's got incomplete. It's got RU5, RU3, and RTR is U3, RU5. You need a full LTR to get virus made, and the RNA is not good enough to do that. So that's why we do all these weird primings near the 5' end, template exchanges to get a DNA with 2 LTRs. So you have an LTR on the left, you have a primer binding site, a PPT, a polypurine tract, and an LTR on the right. And this is going to integrate, and both LTRs are going to contain transcriptional promoters and transcriptional terminators. And if you want to see an animation of this, I made one a couple of years ago. It's on the YouTube channel. May make it more clear for you. All right? So let's see if that made sense. Next question. Which of the following steps occur during reverse transcription of retroviral genomic RNA? Priming of minus DNA synthesis by tRNA, 2 template exchanges, degradation of viral RNA by RNase H, generation of 2 LTRs, all of the above. Let's get the answer. And Mark was right, all of the above. And well, that's what these quizzes are for, to see how you're learning. Degradation of the RNA by RNase H certainly occurs. Generation of 2 LTRs, that's the most exciting thing to me. You go from none to 2. So here's a summary of this process. We have our DNA, well, what we're going to talk about next. This is our product that we have just made by reverse transcription. 2 LTRs, the coding region of the genome, and another LTR at the right-hand side. And this is going to integrate into a host target. So the host is going to be shown in purple. And then you're going to end up with proviral DNA, which is integrated. You're going to have host DNA on the left, host DNA on the right. You are going to lose a couple of bases of the LTR. Look, we got TG. So we've lost AA. Same thing on the right. And we have duplicated a little bit of host DNA. The red and the orange is duplicated host DNA. You'll see in a moment why that's so. This is going to be transcribed by host RNA Pol II because there's a strong promoter in the LTR. In fact, there's a strong promoter in each LTR. And so think of the consequence when this DNA integrates next to a gene in the cell that's normally silent. It could turn on the transcription of that gene. Remember that. Keep that thought. But the transcription is going to give rise to the viral RNA genome. Capped, polyadenylated, but no LTRs, just pieces of the LTRs. That's why they have to be regenerated during reverse transcription. A few words on integration. Right, the reverse transcription occurs in the cytoplasm, then imported into the nucleus, where the integrase protein, which is this beige protein here, the big one, is going to mediate integration of the viral DNA, which is this circle, into chromatin of the cell. And there is a protein in the cell called BAF, which prevents the viral DNA from integrating into itself, because it would do that if it weren't coated with BAF. It gets in the nucleus. Then there seems to be some affinity of the integrase for these BRD proteins, bromodomain containing proteins, which bind acetylated lysines in histones. Remember, histones can be acetylated to regulate their tightness of chromatin. They attract integrase. So acetylated regions may be preferentially the site of integration. And here's how integration occurs. Integrase IN is a tetramer. It binds to the retroviral DNA, binds to the ends. It takes off 2 bases from each end. You can see here we've taken off from the 3' end 2 bases here and here. And then we have our host DNA in purple, which is also bound to the integrase. The 3' hydroxyls newly generated by this trimming attack the host DNA and are ligated to the host DNA. As you can see, both strands are ligated, the dark blue and the light blue strand. And then that's shown again below here without the integrase. And then the host proteins repair these gaps. So there is a gap there that has to be filled in. And you can see in red and orange it's filled in. And that's the duplication of host DNA. You have a gap, so when you fill it in, you're copying the other strand, and that is by definition a duplication. All right, so where does this integrate? It seems to integrate at many sites on both strands. It's not random, but there are a lot of different sites. So here is a table which says what would be the percent if it were random within genes or at transcription start sites. So it would be 26% and 5%. And what are observed for three different retroviruses. And you can see the numbers don't jive, right? They're higher than randomness, even with respect to transcription start sites. So there is some specificity for integration, but it's at a lot of different sites in the genome. All right, so the summary is here we have one DNA produced from 2 RNAs by reverse transcription, right? There's a strong promoter built during RT, which is in this LTR. Once the DNA is integrated, it directs transcription of the host machinery, transcription machinery, to make many copies of viral RNA. And then the viral RNA, of course, is translated into proteins. So as far as I can see, there is no viral DNA replication, and there is no viral RNA replication, as we have talked about for other viruses, right? Put 2 RNAs into one DNA, and you integrate that in the host cell for transcription. So it's unique because you don't have those 2 major processes going on. Now, of course, if the provirus is integrated, of course, as the cell divides, you can get more proviruses as well. But it's not really DNA replication where you're making lots of copies from a single genome. So really unique process. So there's the summary. The reverse transcription in the cytosol, the DNA is imported, integrated into the cellular DNA. You make a messenger RNAs, which are either unspliced or spliced to give structural proteins to make new virus particles. And I said we'll explore that next time in more detail, the assembly of virus particles. Now, sometimes these proviruses don't just integrate into your somatic cells. You know what a somatic cell, right? Everything but your germ cells. Sometimes they integrate into germ cells, sperm or egg, and it gets passed on to offspring. And once DNA is in, it can't get out. There's no mechanism to get it out in the cell. And so our genomes are full of proviruses from ancient retroviral infections. And they're there. We have hundreds of them. They're called retroelements because they were made by reverse transcription. If it's put there by a retrovirus infection, we call it an endogenous retrovirus, because endogenous means it's integrated into the germline. So you can imagine many, many thousands of years ago, you have an individual who is infected with a retrovirus. This could be Neanderthals, our ancestors, right? They get infected and integrated in the germline. They then pass that endogenous retrovirus onto more people, and it passes through the population. And it can take thousands of years for this to fix in the entire population, depends on the size of the population. And some people will have the provirus forever, and some people will lose it. Humans now have lots of proviruses in us. I'll show you in a moment. Sometimes, depending on the animal, so if this is humans, most of the time during the evolution, mutations get fixed in these proviruses and they make the virus non-infectious. So we don't make any infectious retroviruses from these endogenous retroviruses, but other species do. Mice have endogenous retroviruses and they make virus. Endogenization. In us, it happened hundreds of thousands of years ago, but it's happening today in koalas. So we can watch it actually happening. Koalas, of course, are native to Australia. If you see a koala in New York, it's coming from Australia or was bred here for a zoo, right? But they're native to Australia. 50,000 years ago, there was a transmission of a retrovirus from a rodent to a koala in Australia. And that virus has spread through the koala population. We know this because it immunosuppresses the koalas and they get various infections and die. So people are very worried about this. If it were neutral, we would never know this. But this is a cool map showing you in different parts of Australia the fraction of koala populations that are positive for these endogenous retroviruses. You can see in some parts of Australia, they're quite high. In other parts, they're lower. A particular island, Kangaroo Island, has only a few. But these are all becoming positive with time. So people are trying to find koalas that are virus negative and keep them separate so you can have a proper breeding style. But this is an opportunity to study endogenization. Now, retroelement, which I've used before, the definition of this is a sequence that moves in a genome by reverse transcription. All right, so how does that happen? So we have an integrated element from an endogenous retrovirus, right? And it can be copied into mRNA. That can be copied into DNA by reverse transcriptase, which we know is encoded in the nucleus, and then it can reintegrate somewhere else. So it's moving around by virtue of reverse transcriptase. As I said, proviral DNA in the germline is endogenous retroviruses. In humans, all of these endogenous or retroelements are defective. But look at this. 42% of the genome of humans comprises mobile elements that move around by RT. And this includes endogenous proviruses, which would be put there by an infection with a retrovirus, and other retroelements. So here's the breakdown. We have 2.7 million retroelements in our genome, 42% of our DNA. And those are the different kinds on the left. So let's go through those. So on the top is an endogenous retrovirus. So about 7% of our genome, 8% of our genome are endogenous retroviruses, which means remnants of previous retroviral infections, okay? Then we have retrotransposons, which have 0.6%. These are probably the progenitors of retroviruses. So they have LTR and GAG and POL, but no envelope. So these can't make virus particles. By the way, the endogenous retroviruses have LTRs. They have duplicated host sequences, which tells you that it was an integration mediated by integrase. Retrotransposons don't have envelope. So we think these existed before retroviruses, and somehow they captured an envelope from a cell and became retroviruses. So retrotransposons have been around for a long time, but they do have reverse transcriptase. You can see POL. And then we have non-LTR retroelements, quite a bit of those. They include lines. So lines have they have no LTRs, but they do have open reading frames that have reverse transcriptase activity. And then we have two forms that have no reverse transcriptase, where these are just cases where mRNAs were made and copied into DNA and then reintegrated. So we call these processed pseudogenes or short interspersed nuclear elements. And they comprise quite a bit of our genome. So of these LTR-containing elements, less than 0.05% are active in the human genome today. But there are wonderful examples of the use of these retroviral proteins for our biology. And syncytians is a great example. So the placenta is made by having an outer layer of fused cells called syncytiotrophoblasts, ST, that's the red here, to divide the maternal and the fetal circulation. So here's the maternal blood on the outside, the fetal vessels are here. These are very important for keeping the two separate. Those form due to a protein called syncytin, which is a former retrovirus envelope protein. So the envelope protein, as you know, allows the virus to fuse with host cells. So if the envelope is just expressed on a cell, it can fuse with the neighboring cell, and that's how syncytial trophoblasts arise. This is called exaptation, when you take, say, a viral gene and use it for something else for your purposes. There have been multiple exaptations of syncytians for mammals. There are at least two shown here, 50 million years ago and 25 million years ago, in different lineages here, ancestors and so forth. This allowed the placenta to be made. Without syncytian, we would not have placentas, we would not have live births. I don't know, maybe we'd be kangaroos, marsupials. Do we have we don't have marsupials on there. Or maybe we'd lay eggs. I don't know. But we would not have placentas without retroviruses. So you can thank them. It's an old infection, many, many years ago, millions of years ago, and we inherited it from ancestors, but that's what they do. Embryonic development seems to be influenced by retroviruses. There's one endogenous retrovirus called HERV-K, which infected our ancestors about 200,000 years ago. So here's 200,000 years ago. So here's Homo sapiens, and there is a descendancy from Homo rhodensiensis. They're the Neanderthals. So somewhere around there, we're infected with HERV-K. We do not today make infectious virus. These endogenous retroviruses are in us. But mRNAs are made during embryogenesis, from the 8-cell stage to the epiblast. So 8-cell morula and epiblasts all make this HERV-K mRNA, and it induces an antiviral protein called IFIT-M1. So this is what I was mentioning before. This endogenous retroviral protein induces an antiviral protein, so maybe it's protecting these early embryos. Okay, why are chicken eggs blue? So chicken eggs can be multiple colors, and if a farmer has a chicken that suddenly gives rise to a blue egg, which is this one here, it begins to breed this, so it makes lines that make only blue eggs. What is making the egg blue is an endogenous retrovirus inserted next to the gene that makes blue pigment in the uterus of the chicken. So the retrovirus is driving, from the right-hand LTR, it is driving this pigment, which would normally be silent, and so that's how you get blue chicken eggs. So your eggs, yeah, they have endogenous retroviruses in them, but it's okay. It's certainly better than H5N1, which is, as far as we know, not in eggs. So without retroviruses, we'd be laying eggs and they would be white. Amazing, right? Another question here. Last one. Which of the following statements about retroelements is not correct? There are many copies in eukaryotic genomes. They are currently entering the koala germline. Those in the human genome produce infectious viruses. They can be beneficial, none of the above, which is wrong. What is your answer?
My answer is C.
Yep. Yeah, in humans, the endogenous retrovirus do not make infectious viruses. They can make mRNAs and proteins, but no viruses. You know, a number of years ago, someone rescued one of these endogenous retroviruses in the lab. They corrected the mutations and made virus. People got a little upset about that. I didn't. I think everything is good in science as long as you do it safely. All right, the last virus today with reverse transcriptase, the hepatitis viruses. This is cool because these are viruses with a DNA genome, right? Hepatitis virus has a capsid inside of an envelope. The envelope has glycoproteins in it, of course, and within the capsid is a double-stranded DNA, which I have shown you many times to be broken, right? It's gapped, it has a protein stuck on one end, and it has an RNA stuck on the other. Today, I know you've been dying to learn this. Today, you're going to learn why they are there. I know you're not dying to learn. It's okay. I'm not offended. But that protein is reverse transcriptase, actually. So that's a clue for what's going on. So these viruses, they bind cell receptors. The nucleocapsid is deposited in the cytoplasm. It docks on a nuclear pore, and the DNA goes into the nucleus. What happens next? It's repaired by the cell, right? And then it's chromatinized because the cell doesn't like this foreign DNA in there, and so it chromatinizes it to shut it off. And that closed circular DNA can then be transcribed to messages to make new virus genomes and new virus particles. 2 points here that are really important. There is no genome integration here. Not needed. Doesn't happen. From that, you could predict whether there's an integrase or not, right? There's no integrase because you don't integrate. So that's one thing. The other thing, you build a new virus particle. Let's see. We've got some capsid proteins here. We've built a virus particle, and you've put mRNA in it. That's the genome RNA, the full-length transcript of the double-stranded DNA called pregenome. It's packaged into the capsid along with reverse transcriptase. This virus cannot wait to start reverse transcription. It's just dying to reverse transcribe. It doesn't wait till the next cell like retroviruses do, right? Retroviruses bind the next cell, another cell, and they reverse transcriptase, do reverse transcription in the cytoplasm. These viruses started in the cell in which they're being assembled. Really anxious viruses here. The problem is the capsid is closed at this point, and it runs out of dNTPs. So that's why there's a gap in the product. There's no more dNTPs because this is a closed capsid, and dNTPs can't get into it. So that's why all the genomes have a gap. Why they have a protein and an RNA, you will see in a moment, okay? So now you know why there's a gap, right? You get it? There's your gap. Reverse transcription is remarkably similar to that of retroviruses in that you have template jumps, priming at weird places, and so forth. So here's the viral RNA at the top here. There is the reverse transcriptase. It's bound to a stem loop near the 5' end of the genome, right? There's the 5' cap, there's the 3' poly A sequence. So the reverse transcriptase begins to copy the sequences within this stem loop. And then the next sequence of events, there's a template exchange where that product goes to the end of the genome there, and the reverse transcriptase copies that. So there's no more copying of the 5' end here. There's no LTRs here to be built, by the way. So here's the DNA being made. You eventually make a full-length copy of the RNA, which would be a minus DNA. And there's an RNA primer left at the end of this DNA. And that serves as the primer for the second strand, the plus strand of DNA, which you can see starting here. But that's near a 5' end also, so that's a problem. And eventually the DNA reaches the 5' end. There is a terminal redundancy in the genome, so this DNA is able to make a template jump to the 3' end, and that's how you get copying of the other strand. So there are a multitude of terminal redundancies and so forth that mediate this, which are not so important for you to understand. What I want you to understand is that this process stops at some point with a gap because there are not enough nucleotides in this closed capsid. And so you end up with the DNA that has a gap. So you can see if this stopped here, you would have this gap. You'd have a piece of RNA still present. That's the primer for that strand. It was never removed, which normally it would be. And then the reverse transcriptase, which is in the process of copying the RNA into DNA, it stops cold. It doesn't leave the template. It just stops, and that's why the reverse transcriptase is there. RT, primer RNA, and gap. All because this virus is too excited to get things going here. As soon as the RNA gets in the capsid, it begins to copy it into DNA, and you never finish it. But it doesn't matter, because when it enters a new cell, it's fixed by cell enzymes. So you could be a little bit sloppy as a virus, and things still work out. But this is interesting because it's reverse transcription, yet what's in the particle is DNA, not RNA like the retroviruses. And the sole reason for that is because the reverse transcription commences in the producer cell and can't finish. If these viruses were released with RNA genomes, then presumably they would do reverse transcription in the recipient cell, but that's not how it works. All right, so here's the summary, what I just showed you. RNA genome copied to make that first strand. There's a template jump, and then you never complete the cycle. So how does this fit into the overall virus world? Today we have talked about retroviruses, and the yellow box means what's in the virus particle. So for retroviruses, the RNA is in the virus particle, right? You make the particles in a cell, reverse transcription doesn't begin until that particle infects a second cell. So you make LTRs with the proviral integrated DNA, and then you make new viruses from that. The hepatitis viruses, which we've just talked about, have DNA in the virus particle because the reverse transcription happens in the producer cell. And then when this infects a new cell, it's repaired by the host enzyme. But again, this remains this way because there are not enough triphosphates in the capsid, in the cytoplasm, to finish reverse transcription and remove those proteins and RNAs. There are actually two other kinds of viruses with RT in their reproductive cycles. On the top here is cauliflower mosaic virus. And these viruses also package DNA. They have RT in their reproduction cycle, yet they package DNA, and that's for the same reason that hepatitis B virus does. They start reverse transcription before they leave the producer cell. And they also have problems with their genome. They have gaps and they have RNA pieces attached. They get fixed when the virus infects a new cell, and it becomes nicely closed circular, from which new RNAs are made to resume this activity. Now, most retroviruses have RNA in the virus particle. So these cauliflower are a different family. They're not retroviruses. But a set of retroviruses called foamy retroviruses. They're called that because they make infected cells look foamy. They package DNA. 5 to 20% of virus particles actually package DNA. Again, because reverse transcription begins in the producer cell. So you can see a hybrid of this reproductive strategy in these different viruses. The retroviruses are the purists. They put RNA in and they don't make DNA until they get into a new cell. But the other viruses, we have DNA package, and here we have a mix of the two. So the foamies can package RNA, but sometimes this is reverse transcribed in the producer cell so that you have DNA package. But it doesn't matter really because it all works. The ones that package DNA are fixed in the host cell and infection can proceed. All right, next time we're going to put together some virus particles.